Hello everyone. I am Dr. Sutapa Datta working as a scientist in TCS Research, India. Today I am going to present my poster entitled Importance of Genes and Pathways in Deeper Understanding of a Disease, a comparison study using deep learning. My co-authors are Dr. Thomas Joseph and Dr. Rajgopal Srinivasan. We all know that gene expression has been used vividly in different machine learning or deep learning methods for the last couple of decades. And with the advancement of AI, especially explainable AI, it is now possible to identify important genes involved in disease diagnosis, prognosis, and therapeutics. But it has also been observed that these causal genes mostly vary even within patient cohorts and with experimental setup. But this diverse set of genes often converge to a common set of pathways through pathway enrichment studies. Here we hypothesize that considering both genes and pathways in a ML or DL model can lead to better understanding the biology behind a disease. Most of the existing methods, except couple of methods, use only gene expression data. Here to evaluate our hypothesis, we use PNET, a deep learning based framework to identify important genes as well as pathways to differentiate between neoplastic versus non-neoplastic cells or tissues in different cancers. To establish our hypothesis, we have used both single cell and bulk RNA-seq data from three types of cancers. First one is glioblastoma multiforme or GBM, for which two different single cell datasets taken from GEO. Due to the lack of samples in TCGA for GBM, we couldn't use bulk RNA-seq data. Second one is prostate cancer, for which one single cell data from GEO and one bulk RNA-seq data from TCGA are taken. Third one is breast cancer, for which also one single cell data from GEO and one bulk RNA-seq data from TCGA are considered. All TCGA data are obtained using recount2 bioconductor package. The study is done using PNET architecture to differentiate between neoplastic versus non-neoplastic cells or tissues. PNET is a deep learning based method which uses known genes and pathway information from reactome in multiple layers to originally classify primary versus metastatic prostate cancer samples using mutation and CNV data. Deep lift method integrated in PNET obtains the important genes and pathways which contribute mostly in the prediction process. This is the PNET architecture that we have used, and this is the block diagram of our analysis where transcriptomic data is used as an input in PNET to predict neoplastic versus non-neoplastic cells or tissues. Top rank pathways and genes obtained from PNET are then verified using cake disease database and literature survey. Here comes the result section. This is the confusion matrix for GBM, and this table shows the prediction performance of PNET for all the datasets. PNET yields a high value in terms of all the statistical parameters like accuracy, precision, recall, AUC, F1 measure, AUPR for all the datasets. Next is the confusion matrix for prostate cancer. Table shows PNET gives a high performance for bulk RNA-seq data, but it yields an average performance for single cell RNA-seq data. This may be due to some artifact in the dataset or experimental setup. Here two single cell RNA-seq datasets are taken from organoids and paired samples with same GEO accession number. Next, result shows the confusion matrix for breast cancer, and the table shows that PNET performs very well in terms of all the statistical parameters for both bulk and single cell RNA-seq datasets. The next table shows the top 10 genes and top 5 root pathways based on the coefficient value that we retrieve from PNET using deep lift method. For GBM, we got genes like EGFR, GNG5, which have established role in GBM according to literature survey. And looking at the root pathways, we got some common pathways like cell cycle, extracellular matrix, etc. Cell cycle is a very prominent pathway in almost every type of cancer. Although the gene varies with different datasets for GBM, we got some common pathways. Similarly, for prostate cancer, bulk and single cell datasets, we got a different set of genes, but those genes converge to a set of common pathways like cell cycle, metabolism, neuronal system, etc. Here also cell cycle appears to be a prominent pathway. Similarly, for breast cancer, both bulk and single cell RNA-seq datasets, some gene appears to be from same group like RPL and RPS along with some prominent gene like CDK1. but they mostly converge to a common set of pathways like cell cycle, gene expression, immune system, etc. So for all the data sets, cell cycle appears to be a very prominent pathway across all the cancer types. This table shows non-root pathways obtained from PNET which are found to be in cake disease database. Here also several pathways are seen to be common across datasets for a particular disease like meiosis, cell cycle checkpoint, mTOR signaling by WNT for GBM. Similarly for prostate cancer, RNA polymerase 1 and 2 transcription signaling by nuclear receptor, cell cycle checkpoint, mitotic G1 S phase are common across different datasets both for bulk and scRNA-seq data. For breast cancer also, pathways like apoptosis, meiosis, cell cycle checkpoints, M phase are common across bulk and scRNA-seq datasets. Here also, cell cycle related pathways are common irrespective of cancer types. So, in conclusion, this study demonstrates the need of pathways in a model along with genes as a means of better understanding the similarity across patients and data types in an automated way. Also, this type of model will help in better understanding of disease biology and design of therapeutics that work across most patients with same disease. These are some of the references we use for our study. Thank you.